Hello and welcome. Good afternoon, good evening, good morning, wherever you're tuning in from. My name is Sam Ankerson. I'm director of the Bailey Matthews National Shell Museum, and we're delighted to have you for this program with Dr. Jann Vendetti, Land Snails in Los Angeles, an experiment in urban citizen science. Jann is a terrific asset to our field, Malacology. She's the curator of one of the most important collections of mollusks in the country, housed at the Natural History Museum of Los Angeles County. She's a great communicator on the subject of mollusks, and has a real way of getting people interested in the topic. And the program she'll be talking about tonight is an inspiration. It's an innovative idea. It is a great venture in citizen science, and is actually has inspired our museum to launch a similar program. It's inspired other organizations and museums around the country as well to do something similar. So you'll hear more about it, but we're just very excited that that Jann is with us for this program. So thanks, Jann. And shortly we will get to her talk. First, just wanted to touch on a couple points about this program and some upcoming ones. If you have questions, please ask them at any time. The way to do that is to use the chat function. So if you're looking for that, if you take your cursor and scroll along either the bottom or the top, the bar should appear, and you click on chat and type in your question, and I'll keep track of those. And after Jann's talk, we'll do Q&A. Some upcoming lectures. So this is the first in our 2023 season lecture series. Next month, February 15th, Tina Petway, the curator of Malacology at the Houston Museum of Nature and Science, will be talking about their big new reinstallation of the shell collection there. March 23rd, Alexa Elliott, who is producer of the PBS program Changing Seas, will be sharing some of their adventures in filming in oceans all over the world. On April 20th, Pete Malinowski, who is the CEO of an organization in New York called the Billion Oyster Project. They are undertaking a very groundbreaking restoration conservation initiative to repopulate New York Harbor with over 1 billion oysters. And on May 17th, our own curator, science director and curator Jose Leal will be giving a talk on micromollusks. So you can find out more about all these programs and sign up for them. They're all free at shellmuseum.org. And also at the same place, the same website, is an archive of all previous online lectures. So there's a great bank there of speakers and topics dating back a couple years now. And this program will also be up there. Usually takes us a few days, sometimes as much as a week to get it on the website, but you can find it there. And so before we go to Jann's program, I wanted to give a brief update on the museum for those here for this program who may not be aware. This museum was very heavily impacted by Hurricane Ian in September, and so I just wanted to give an update and share a couple of slides briefly before we introduce Jann. Let me see here. So here is the museum following the storm. As we can see, it's sort of battered and tattered, but still standing. And I won't go through a whole lot of damage photos, because folks who may have been at our last online lecture, which was in November, on the subject of hurricane and mollusks, we did a lot of that then. But the main events were 5.5 feet of flooding on our ground level. And this is a picture, for those who do know the museum well, of our founding executive director, or a picture of a bust of our founding executive director, Tucker Abbott. And you can see some arrows here on the water line, just indicating how high the water was. But the museum is basically on, it's got two parts. On the ground level are aquariums, about 10,000 gallons in 11 aquariums that exhibit living mollusks. And then up on the second floor are the shell exhibits and a special exhibition gallery. And in November, we looked at pictures of a lot of bad flood damage. The state of the museum now is that it's basically about 40% of the museum has been gutted. So that means that its walls, its ceilings have been torn out to free the place of water damaged and moldy surfaces, and make it possible to rebuild. Here's down on the aquarium level, can get a sense of sort of what's still there and what isn't. The aquarium structures themselves seem to be okay. We are doing a little bit more testing to make sure of that. We salvaged as much aquarium equipment as we could. So over here are some examples of that, things like filters and pumps and pipes and all the kind of back of house systems that make the aquariums operate. And we've been testing those and are finding, unfortunately, that most of them are not in great shape. Some will be able to salvage, but most not. Right here is the giant Pacific octopus's aquarium, and as folks who have tuned in before, have been paying attention to what's been going on in the museum, no, we unfortunately lost about 75% of the marine life in the storm. So moving up to the second floor where our shell exhibits are, this is the Great Hall of Shells, about a 3,000 square foot space with 35 different exhibits of shells, and it suffered damage as well. So we didn't just get flooded on the ground level, but because of impacts to the roof, we had water coming the top as well. So you can see this space is gutted, and then down closer to the exhibits themselves, not a whole lot of that area was impacted in this way, but maybe about 25% down, lower on the Great Hall's exhibit level had this kind of gutting, either walls or ceiling that needed to occur as well. So it's a lot of remediation, as we say, but the good news is after about 2 months of this kind of work, the museum is now it's now dried out. It's clean. It's free of humidification problems, and has been sprayed for antibacteria and anti-mold. This is up on the third floor, actually, where another place where we took roof damage is it's a collection storage area, actually, not where we exhibit shells, but the scientific collection of over 500,000 specimens. And this is where the space we're looking at here, which is obviously empty, but is where about 30% of the collection was stored, and you can still see the remnants of the hole that was torn in the roof. It was a much larger area. It's now temporarily patched. But that area is remediated as well, and we located that part of the shell collection, which was in harm's way, down to a part of the second floor, which is unimpacted. So we're ready for rebuilding now. The emergency response phase of things is over. We've received bids from general contractors. We're also working with vendors that specialize in aquarium design and installation, and also exhibit designers, as would relate to the Great Hall, to integrate all of that into to start work. We don't know exactly when it's going to begin, but we do know it is going to be this spring, which is good news, because there was certainly a fair amount of time in the fall where the assumption was that with all the rebuilding work that needs to happen around here that it would be hard to get any work started, for many months. But it looks like we may be able to begin sooner than we had thought. How long it will take is probably it's too early to say something with any confidence, but we're looking forward to getting started soon. And until we do, we are, and even after we do, we're glad that we've also been able to restart or launch new educational programs. One of those is an outreach school program called Mollusks on the Move, where our educators take live mollusks and shells to schools around Southwest Florida. And you can see on the right, there's two of our educators getting ready to do that. We restarted that on January 4th, and we're visiting 32 schools between January and May. We will actually be, believe it or not, reopening the museum for a kind of limited edition experience on February 1st. In about 10 days or whatever that is. It'll be free admission, but until reconstruction begins in earnest, visitors will be able to view the Great Hall of Shells. We're putting together a temporary exhibit, and on the left, this is a photograph from it, called "In the Wake of the Flood: Community Photographs of Hurricane Ian and its Aftermath," where we're inviting community members to submit any photos they have that are meaningful to them about the storm or its aftermath, good, bad, whatever the subject may be, and we'll exhibit all of them without any curation going on. So looking to get a collage-like community perspective of the storm. So that will be up to view free of charge, and we'll stay open like that until reconstruction begins. So we're looking forward to having a opening up so the public can come in again. And finally, I mentioned we're as a museum, we're inspired by Jann's work in land snails and slugs and observing them in the field. We're kicking off a program actually this coming Monday called Snail Search of Lee County, where we basically follow a model like you'll hear about from Jann, but the goal is to get out into the field with staff, with volunteers, and mobilize people throughout Lee County to get out in the field and do wildlife observations, and in particular look for species of land snails that are out there, and to document that using the app by Naturalist that you'll hear more about. So that's an update. Just before I'll introduce Jann just a second, but just thank everyone for who may be on this Zoom, who has been paying attention to what's been going on, who's maybe supported the museum during this time, offered moral support, offers to help, financial support, the whole thing. It's all very appreciated, and of course we've still got a long road and there's more to come, but thank you. So and now it's my pleasure to introduce Jann Vendetti, who is the Associate Curator of Malacology at the Natural History Museum of Los Angeles County. She's the co-director of the museum's Urban Nature Research Center. She joined the museum in 2014 and has focused on collection-based research and the Snails and Slugs Living in Metropolitan Environments SLIME project. She earned her PhD from University of California, Berkeley's Department of Integrative Biology and Museum of Paleontology, and had a postdoctoral fellowship at California State University. And she's just a terrific person, great malacologist. And Jann, thank you and welcome.
Thanks so much, Sam. Can you hear me okay?
Can.
Yes, and you can see my slides?
Can see the slides.
Wonderful, thanks. Thanks so much. Thanks for that introduction. Thank you for inviting me. And I'm also really glad to hear the progress on the museum. So that's all good news. Thank you all for joining. I'm really excited to have this opportunity to discuss this project. Land snails in Los Angeles, this experiment, or an experiment in urban citizen science. At the museum, we also call citizen science community science, although there's some debate over the difference between those terms, but sometimes we'll use community science as well. let's see. I just would like to start off by saying this quote and having you see it. The primary goal of natural history museums is to preserve a biological record of the natural world for scientific study. This was Joseph Grinnell's statement many, many decades ago, and something that is of primary importance to all museums and certainly natural history museums. So what is important to us and important to me as a curator and important to the Shell Museum is to have these specimens as a biological record of biodiversity. And there are creative ways to think about how one can, as a museum or another institution, also gather data as biological records of the natural world for scientific study. And so that's where this community science or citizen science program comes in. This is the Natural History Museum of Los Angeles County. This is the old entrance that was built about 100 years ago. The museum was established more than that. In 1913, it has about 35 million objects and specimens and 15 organismal departments and collections. And we think of this as a biodiversity collection, of course. Sometimes I call this a library of biodiversity, just for people who are unfamiliar, that instead of books, all of the specimens that we have are, all of the books are the specimens are our books. That's a little clearer.
Hey, Jann,
yes,
sorry to interrupt.
That's okay.
There's been a couple questions whether there's a way to make the slides full screen.
Oh no, I haven't. Yes, of course there is. Yes, there is that full screen.
I don't know. I'm not on mine. I don't think.
Maybe someone in the chat can say. I think that.
Yeah, maybe someone in the chat can say. Are they?
Oh, sorry, everyone. Yeah, I should have put it in slide mode. Yep, I think that that's better. Okay. Thanks. Thank you for pointing that out. Okay, so our collections look like this. They're not on display in any appreciable way, unfortunately, but the primary reason for our institution is to have these collections as scientific specimens for scientific study. So they look like this. We have a number of locations on-site and off-site where our specimens are kept. We have a number of specialties in our specimen collections. But that's what they look like. And then you all probably know this, but just to recap, the mollusca are incredibly diverse as a group, only second to insects in species diversity. And there are about 100,000 species, maybe 75,000 to 100,000, and about 40,000 of those are land snails. So the greatest proportion of those mollusks are gastropods, and of that 40,000 are land snails. And to put that in perspective, I usually tell people that just to get your mind around those numbers, that there are about 15,000 to 18,000 species of birds, and there are about 10,000 species of mammals, and yet there are 40,000 species of land snails. So it's a tremendously species-rich group, and one that's kind of hard to get your mind around if you haven't studied them before. And then just within all of the mollusca, these are our gastropod friends that can come in a number of different forms, with shells, without shells, as slugs, as snails with shells. And of course, they're related to the rest of our gastropod friends, the octopus, cuttlefish, chitons, mussels, etc. Okay, so at the museum, I became associated with the Urban Nature Research Center. This is what our landing page for that program looks like on our website. That's my colleague Miguel, who is sort of famous for P22, the puma or mountain lion that until very recently lived in Los Angeles, the backyard, if you will, of Los Angeles. Anyway, this group of curators and associated scientists at the museum made a push to particularly study urban.
Jann, I'm sorry to interrupt
okay.
again. The slide we're seeing is still the collection cabinets.
Oh, what?
Yeah.
Oh my goodness. Okay, let me stop sharing and reshare. Oh my goodness, I'm so sorry. Why is this happening?
There we go. And now we're full screen.
Okay, great. So what are you seeing now? Are you seeing mollusks?
mollusca,
Now you
100,000
see,
species.
now you see
Yeah.
a bird and some mammals.
Yeah.
All
Yeah.
right.
Yeah.
Oh my gosh.
All right. We're
sorry.
good. We're good. We're good.
Thanks, Sam. Okay.
Thank you.
So, so this is like I was saying, this is my colleague Miguel, and the Urban Nature Research Center is a group of associated scientists and other staff members at the museum who made a concerted effort to focus on urban natural history and urban nature very specifically. We are in a giant county of 10 million people and 503 square miles, and it ranges from the Mojave Desert all the way to the ocean. And much of the population density is focused in the city of Los Angeles, the biggest city on the West Coast. So where the museum is located. So we decided to focus our research efforts instead of someplace that to us would be exotic or far off. Instead of doing that, we would focus our efforts in our neighborhood, in our county, in our city, and in greater Southern California. And then from just a personal perspective, my training and my interest in natural history and biology was inspired by tide pools, East Coast tide pools, in fact. And I had to switch my sort of passion for finding and being excited about invertebrates in this particular environment to a terrestrial one. And I thought of it as leaf litter. Moving aside seaweed is almost the same as moving aside leaf litter. And so I started to get as excited about the biodiversity in urban environments or anywhere else on the ground, in rotting logs, in leaf litter, etc. So for us in Los Angeles, that's where most of our snails and slugs are found. So it didn't take a lot to make that mental shift, and it became a really exciting project for me to move from the marine environment, at least for this project, into the terrestrial environment. So as somebody who was unfamiliar with land snails and slugs, I was a professional malacologist. Nonetheless, I said, well, let me get the field guide and learn and figure out what species there are. So I knew what I was doing. These are California natural history guides. I'm not sure if other states have them, but there are many. There's one to just plant galls, owls, birds, mammals of California. And so if I were to have one of these for mollusks, I would really be starting off on the right foot. Actually, this doesn't exist. So there is no snails and slugs of Southern California. I just faked this by adding a different title to birds of Northern California and a different image. So I was sort of stuck in not having a great place to learn all of these taxa, although there's quite a wealth of information by Pillsbury's work in the 1930s, where he documented much of the biodiversity of land snails and slugs in all of North America, but something that was a little easier to get my head around. So that was part of the inspiration for developing an iNaturalist project. So as Sam mentioned, the project's name is Snails and Slugs Living in Metropolitan Environments, or SLIME. And the iNaturalist platform was just beginning to become very, it was getting some of its bugs out, and it was becoming very user-friendly when we started the program in 2015. And so this project, the SLIME project on iNaturalist, was modeled also after my colleague in herpetology, his project, which was called RASCALS, which is another acronym, and that documents species of reptiles and amphibians, also at the museum. He's a curator at the museum. So we started SLIME, and in a couple years, we had almost 17,000 observations. If you were to join iNaturalist, so iNaturalist, the sort of simplest way to put it is it's a online biodiversity platform. You could think of it as like Instagram for biodiversity. So you post an image of any living organism or dead organism, pardon me, and there's an algorithm that gives you a suggestion as to the species identity, and that algorithm is learning. So it gets better and better the more people use it. And so you can get very good suggestions for the biodiversity, for the species that of the image that you took. So it might say, you have this, and look at this, like this is the species that you found. And or other people can agree with the AI's species identification or disagree. And then there's sort of a thread of conversation that can happen within each observation about that species. So it's a fantastic way to learn about local biodiversity or whatever biodiversity you are interested in, and also make identifications when you get if you have a familiarity with a certain species. So anyway, we started this project, and we had to use a number of museum resources to get the word out. We also started a collection. So the very smallest collection, or specialty collection we had in the museum, at least within malacology, was land snails and slugs. We had almost no slugs, and we had very few land snails, and not great data associated with those land snails. So we grew that collection along with this iNaturalist project. So we did things like social media outreach and media releases. We asked people if they saw these different species that we do not have in Southern California, or should not have, but they would be potentially invasive. So we were asking people on social media if they saw them and to upload their observations to the project. We did this again right after we found the garden Arian, or Arian hortensis, and got a number of observations. This slug is now ubiquitous throughout California. This is the only slug native to Los Angeles County. And I think it's possible this is the first photo of it ever. That's very possible. And it was from iNaturalist. And we asked people to make observations, or to post the observations they had in particular of this slug, because we knew almost nothing about it or where it lived. We also got some coverage in the LA Times, which was great, at one of our events, and we had multiple events. So every couple months we would have an outing, sometimes called a BioBlitz, where we would go to some location, sometimes to a nature center, sometimes to an urban park, and advertise to museum guests that we were going as a group. And then we would look around, and people would use their cameras or smartphones to make observations of anything, but with a special focus on finding land snails and slugs. So this was the LA Times coverage. We also got some coverage from KCET, which is our local PBS affiliate. They did an episode of a show called Earth Focus. And I went into the backyard of Emily and Gregory Hahn. This is Emily right here. And they had two species that were first records in Los Angeles County in their backyard. So introduced snails, not native. And it was this sort of interesting story that they posted one to the project, and then we contacted them, and then this reporter found out and thought it was a good story, so we ended up in this program about it. We also have had a show at the museum called the Curiosity Show, and so that's me and my colleague Chris Thacker talking about land snails and slugs in the collections. And we also did a project every year that every year we have had the slime project. We did a sort of sub project called Snail Blitz, which was 2 months, and it would coincide with our rainy season. We just had our rainy season, like unprecedented rains here, which they came a little too early for the Snail Blitz. That's all right. But typically our rain comes in February and March, and that makes it more amenable environment to snails and slugs, and for them to be more obvious to people. So we coincide this extra publicized part of the slime project for those 2 months, our rainy 2 months. And we make a goal of 1,000 to sometimes 2,000 photos or observations of snails and slugs. And we often get really surprising records, and sometimes new records for counties or state or the state, counties in Southern California, or new records for the entire state. We also gamified it a bit and provided a bingo sheet. This was this year for Snail Blitz. So we distributed these. This was on the website, and a number of teachers used it for school classes, and it had like cute elements and some fun biology And it was a way to engage people in snails and slugs, which most people are familiar with, of course, but most people know very little about, and often people don't think of them as even separate species. So this was a really nice way to show people living in the city and elsewhere throughout Southern California that there is a lot of biodiversity around them. And then we also promoted the project on our website. We had a photo contest and we gave prizes for the best photos, and the best was like rarest snail, best photo of maybe 2 snails. We had a whole bunch of different categories. So our numbers started growing. This is at just a little over 18,500 observations. Most of the snails that we were observing, or people were observing as part of the project, were introduced. So the garden snail, the milk snail, milky slug, decolate snail, those first 4 of the top 5 are all introduced snails. That last one on the list to the right, the Southern California shoulderband snail, is native, but everybody else is introduced. So some of our outcomes with this project, we discovered the first record in LA County of an Ambigo limax slug. This is a Spanish slug. There was one species already known to live in Southern California. We discovered a second, and we used some molecular data to do that, to figure that out, as well as dissections and morphological data. We also found the first record in LA County of a European species called Xerotricha conspurcata. This is a a species found across Europe. It's also introduced, and it does well in other places. So there's this theme that I'll get to in just a minute of various Mediterranean and other species that do well elsewhere in the world doing well in Southern California. The same is true for Florida. I think you all have that issue too. The Gardnerian, like I mentioned before, this was the first record for Los Angeles County. We discovered through the project. We did some molecular analysis too to confirm its identity. This is a potentially problematic an agricultural pest snail called Cochlicella barbara, and or the small pointed snail. It's a big agricultural pest in parts of Australia, in grain crops, and we found the first record of it through this project in Los Angeles and just south of us in Orange County. And what was interesting is we had somebody submitting photos, I think for Snail Blitz, and they sent in the photo on the left for that larger snail that you can sort of see on this grass. And I said, could you zoom in more clearly, or more, can you zoom in on the smaller specks there? Because that looks to me to be this species. And then they did, and it was the first observation of this particular potentially pestiferous species in San Diego County since the 1960s. So this got me thinking, and the UNRC group as a whole, of thinking about urbanization as a homogenizing force or as a homogenizer. So just to illustrate this idea, this is a photo of snails from southern Spain, and there are 3 species in this photo. The ones in yellow are Cornu aspersum, the common European garden snail. The ones in pink are Otella lactea, sometimes called the Spanish milk snail. And the ones circled in blue are Theba paisana, sometimes called the white Italian snail. We also have these 3 species in Southern California, but this photo was taken in Spain. So this is sort of this idea that I brought up just a bit ago, that there is this homogenizing force in biodiversity. Urbanization brings a lot of species from elsewhere to different places, and then the species that do really well stay around, and such that you end up with a suite of species in, let's say, a city that is more similar to another city than it is to an environment just miles away that is a native and undisturbed environment. So just as another example, I use slugs of Britain and Ireland all the time to identify the slugs in Los Angeles, because the slugs that we have in Los Angeles, aside from that one species, are all introduced. So this is the very best guide to slugs that I have encountered, and I use it all the time. But it is of Britain and Ireland because most of the species that we have are European, like these British and Irish slugs. Okay, and so this idea of urbanization as causing homogenization, it's not my idea. People have been talking about it for a long time, but it's sometimes called biotic homogenization. That you can, like in this paper at the bottom of the screen, that you can gain species richness, but not because you are conserving species or you are promoting the return of species that were lost for some reason, but you are increasing biodiversity because you're adding in species from somewhere else and then making those locations, two dissimilar locations, disparate localities, very similar to each other because of their biodiversity. And then I also have thought about urbanization as a facilitator. So in a very similar way that urbanization is causing this homogenizing effect of biodiversity, it's also creating the means for species to be moving from place to place. So along with some members of my lab and students and community members who were citizen scientists or community scientists, we published 2 papers of some of the observations that I just described to you, some of the new records that I just described to you. So this first one is 5 new records of introduced terrestrial gastropods in Southern California discovered by citizen science. This was directly an output or a consequence of the efforts of the Slime Project. And then this one on the bottom, the introduced terrestrial slug, Amigo limex, et cetera, is also the same outcome from a project. And the people who are listed as co-authors, many of them are contributors from the public, community members who contributed really helpful observations. So Xerotrichicons percata, this is just another photo of this species. Like I said, it's European. This is Pupoides albilabrus. I didn't mention this one before. This is found in the US Southeast, and we should not have it in California, but we do. There's Cochlicella barbara, also should not have this species, European species in California, but it's quite established in Southern California and parts of Northern California. Otella lactea is the same. This is the Spanish milk snail. It can be this brown, have this brown pattern on it, or it can be purely white. It's one of the most commonly observed snails in Los Angeles, partly because it's a big snail and quite obvious, and it's not found in the leaf litter. Typically, it's found up on branches, tree branches, stalks, structures, cactus, and it makes it much more obvious to people. So there's a bias there, sort of data collection bias, and people see this snail a lot, which is good for us for some of the research that we did on this snail, which included students of mine going out into the field based on iNaturalist records and collecting. So 7 students went to various parts of LA County and collected snails based on iNaturalist records. So they went to a place called Silver Lake and the Bayona wetlands, San Pedro, et cetera. I also had a colleague that was in Texas, and he collected while he was in Texas. And I collected a little bit in San Diego, a student collected in San Diego. And what we found using molecular data was that there were at least 4 different lineages that had been introduced to Southern California. So this became a quite substantial research interest of mine, that not just what species do we have and where are they from, but how many times did this species, how many times had this species been introduced to California? And could we tell that using molecular data? So we did. So we looked at a number of genes and compared them, and the particular gene sequences or haplotypes of the mitochondrion that we looked at were different between these groups, such that they nested with each other and then separate from each other by location, and which makes us think that it must be that this species has been introduced multiple times to Southern California. And just to bring up, this was Howard Hill in 1941. This is in the Nautilus new records of introduced land snails in Southern California. So this species has been known in Southern California since 1941, this Spanish milk snail. But I just highlight the box in blue. It was suggested that Italians living in the vicinity had imported them from Morocco or Southern Europe, as a shipment of live specimens from Morocco was confiscated here several years ago. So there is some history, and I've been trying to get into some of this history to figure out if this was a sort of just blame this on the new Italian immigrants, or is it true that the Italians were bringing in these snails from their homeland or near thereabouts? And so that's been a sort of interesting side project to look into. They've also, this species in particular is eaten, it's very popular in Morocco, in Marrakesh, and they're boiled and served in a soup, the Spanish milk snail. We also have a species that looks very similar, and it's easy to confuse the two, but this is Theba paisana, the Italian, white Italian snail, and it estivates in huge groups, and it can cover entire structures. In the early 1920s in Southern California, in San Diego County, there was there were a number of funny words, or it was a summer of the invading snail, or like the summer of the encrusting snail. It was this event that happened in La Jolla in San Diego County, where these snails were so numerous that they were it was a huge operation to remove them, and they were thought to have been removed. And in this photo, you can see that the structure right in front of, like, the structure is encrusted with snails, but if you look behind it at the branches behind you, those are not, I mean, the yellow things are flowers, but everything else, those are not flower buds, those are snails. So much of all of these plants are just absolutely encrusted with snails. And this was in 2019. You can see that the observer on iNaturalist said they are everywhere. So this awful year of the snail, summer of the snail that happened in La Jolla in San Diego County happened again in LA County just a few years ago. And so these snails were everywhere and were encrusting structures, not throughout the entire city, but at certain locations. So we collected them. We have them in our collections now. We didn't have any from the 1920s, despite there being malacologists on staff at the museum at the time. Or we certainly didn't have many. And these were probably in the many hundreds of thousands of specimens, or excuse me, of animals. So we collected them from LA County and San Diego County, and I did some research in old newspapers. This is a person, it's a little bit hard to see, but there's a person in the center of this photocopied photo from a newspaper, and they're in like a suit with, it looks like a shield over their face, and they're holding a long like stick. And what the caption says is that this is someone attempting to wipe out the pest horde of white Italian snails by whipping flaming torches over the infested area. So somehow before researching this, I had never heard of this before. So people in Los Angeles don't talk about, oh, did your grandparents tell you about the infesting horde of snails in the 1920s? Like there's no history of that is at that sort of top of mind of people. But yet, so looking into the history through old newspapers was really eye-opening. We would not be able to whip flaming torches over an area infested with snails today for various reasons, but this was in 1935. So the control efforts for this species were ongoing for a long time. And one of the important things about a community science or citizen science project is that because there are many people looking for these species, it is possible, or it is theoretically possible, to remove and control a pest species, a pest snail, much quicker than if you were just a government agency or a malacology department, because there's so few of you, but there are so many people. And those people now, with smartphones, with cameras, with the ability to very quickly make an observation and upload it to this website where I can see it, allows, potentially allows pestiferous species to not gain such a hold on certain areas and be eliminated or controlled much better than in previous years. This is just showing that these snails are sold for food in Italy as well. So and in other parts of Southern Europe. So it is possible that the introduction of this species was because of immigrants, again, immigrants in Southern California in the 1920s. And we did a DNA analysis of these and found that the snails that we collected from Los Angeles County had DNA sequences that were most similar to specimens collected in Malta that were available on GenBank, a data site for molecular sequences. And the San Diego County specimens were most similar to snails collected in Morocco. We don't have any Italian sequences in this data set, so it's very possible that Malta was just the closest to Italy. But if we could have Italian sequences integrated into the study, that would help us a lot, and also help us kind of figure out if it is more likely that the Los Angeles County snails were introduced by Italians. Okay, and then one other way of thinking of urbanization, so in addition to a homogenizer and a facilitator, we also think about it as a filter. So not every organism can live in an urban environment just because of the stresses and stressors that an urban environment causes. And then a nice example of this is Helminthoglypta trascae. So we have in Southern California and in all of California, there's quite a large number of species within this genus. These are also called the shoulder band snails. They're brown, they have a darker brown band. They're kind of inconspicuous. They're detritus feeders. They usually live in undisturbed habitat with downed wood and leaf litter. And they cannot live in an urban environment where they do not have those resources. So they cannot live in a lawn or in a garden. They need to eat detritus, and they need to have thick leaf litter. So an urban environment filters them out. An urban environment does not make a habitable place for them to live. While it does facilitate that for a number of other species that have different habitat requirements. So Helminthoglypta trascae here, these are some iNaturalist observations of this species throughout Los Angeles County. And then this is one of my own, and this was a photo that I took, an iNaturalist observation that I made in 2017 on my way. Those are my keys in the picture here, and that's little Helminthoglypta trascae. This was on my way back from teaching a class. I taught a night class at the community college, and I was walking back to my car, and there was a little patch of dirt next to what was otherwise it was next to a building, and then next to that is an undeveloped hillside that goes up into the San Rafael Hills, which is part of Los Angeles. And so anyway, this little snail's crawling along, and I thought, oh, that's amazing. And I took a photo of it, and I went on my way, and I didn't think much of it. And then years later, I was at the college again, and I found it on the other side of campus in a community garden or a campus garden that had been neglected for like a couple years. And I found it under a wood pile, but the place that I had originally found it had been paved over. So this little patch of dirt was entirely paved the next time I went, well, maybe months after. So this is just to say that there is a tremendous filtering effect of species in an urban environment, because most species, very obviously, cannot live where they don't have resources. And I tell people when they ask, what can we do to bring native snails and slugs to our property? And I would say, well, you would have to go to the woods and like get dirt and go to a natural environment and bring some of that into your yard and also have like rotting logs there. And so most people are not going to incorporate rotting logs into their home landscaping. So anyway, so this is as a filter. This is another specimen from our collections, the same species, some of the Glyptotrascites from 1936. And it was, the locality data says it was found on 24th Street in Hermosa Beach. So in 1936, this species was living on 24th Street, Hermosa Beach. This is 24th Street in Hermosa Beach. By the 1950s, it had been completely developed into houses. In 1936, it wasn't. By the 1950s on, it was all houses with no adjacent natural area. And now, if you look at iNaturalist, the most common species found in this area are all introduced. So there's the garden snail, Cornua spursum, the three-bend slug, that's Amygolimax, milky slug, Daraceras reticulatum, and Drapernaud's glass snail or Oxychilus. So the difference between a non-urbanized area having a native species and an urbanized area having nothing but introduced species is the trend that we see around the world. So our project started, as I mentioned, in 2015, and until October 10th, we had it as what at the time in iNaturalist was called a traditional project, which means that people had to join your project or specifically add their observations to your project. So within iNaturalist, there's a capacity to check some of your observations and then have them go to a particular project. And at that time, we through up to October 10th, 2022, we had just over 18,700 observations and 134 species. And this is in Southern California. On October 10th, we decided to switch it to what's called a collection project. And that means that instead of someone having to know that this project existed, despite all of the outreach that we did, it's unlikely that most Southern Californians know that this project, who use iNaturalist, know that this project exists. So we wanted to pull as many observations as we could from this group of Southern Californians so we could see what the biodiversity, the molluscan biodiversity of the area was. So we changed it to a collection project, which automatically pulls any terrestrial snail and slug to our project, whether the person knew that the project exists or not. So that nearly doubled our number of observations. The number of species shouldn't have jumped. I think that's a couple of marine species sneaking in there, and there's a couple of freshwater species too. But it made our numbers bigger, which in itself is not inherently important necessarily, just the numbers, but the data that come along with them, that's what's important. So now we have much more data to look at introduced species, new records, new species that shouldn't be here, and then also distribution data for our native species. So where the native species are within Southern California. And then just as of today, we have a little over 35,000 observations. And our number of species decreased, but that's because I think I booted some of the marine species out. And this project, in talking to some colleagues, inspired Atlanta slime. So this is a small project by a colleague of mine in Atlanta who's teaching and wanted to have her students work on something that would create a lot of new data, because the snails and slugs in that part of the Southeast are underdocumented and understudied. So she started this project, and we're working together on some publications. Some colleagues in the Pacific Northwest also started a project, and they have a lot of, because it's the Pacific Northwest and they get so much rain, they have a lot of native species and introduced species as well. So we're collaborating on a couple of projects with their Slime Pacific Northwest that began, I think, in 2021, and they already have 37,000 observations. And with that, I'll stop here, and I'm happy to take questions, or I could go back and explain something else, something I didn't explain, or yeah, if anyone has thoughts or questions, I'm happy to take them now.
Thank you. Yeah, thank you so much, Jan. Great presentation. It's there's so much about it that I love from a sort of program point of view. It's kind of the creative ways of getting the community involved, and then how your path, took you along to discover all these really amazing things about the natural history of the area. I had a couple questions, and folks can use the chat function to send in any questions. Actually, Kimberly has a comment. Recommend that you look to San Francisco as a place of introduction of Italian, Sicilian snails, since San Francisco is a port of entry for large numbers of Italian migrants.
Yeah, it's absolutely true. There are some, so this, both, let me see, Otella lactea is found in the Bay Area. So the Spanish milk snail is found in the Bay Area, as well as in, in San Francisco and Palo Alto, and then also in LA, but it's not found in between. So it's not like the snails are moving. They're getting reintroduced to these cities. And you're right, there's probably a history. There could be a history of migrants bringing them separately to San Francisco. They also, and I didn't mention this, they also can estivate, so they can go to into snail hibernation for weeks, and they will stick themselves onto something like a truck or a container ship or the outside of a potted plant, and then they can be moved inadvertently. And it's difficult to tell, and I'm not even sure how to tease that apart, but it's difficult to tell how they're getting places, because it could have been that it was purposeful or accidental previously by immigrants, and then now they're moving around just by accident because the snails are attaching to something and ending up in a new place, and then laying eggs, and they're off and running.
Yeah. Harley has a question.
When you learn that they're in a spaceship, yeah. So I had this happen just earlier this week. When there is a new and potentially invasive species, what are your next steps? So as a museum, we are not really tasked with removing species. We could make efforts to do that in collaboration with government agencies, California Department of Food and Agriculture, or the USDA. But the USDA has certain policies wherein they will inspect incoming shipments and imports coming by plane. They will inspect those for snails and slugs. So if you're getting like parsley from somewhere that's not California, but anyway, you're getting it from Europe, or you're getting it from Asia, or you're getting anything that's a plant from somewhere, there will be a small inspection of those goods to see what snails and slugs they have in them. And if it's a species that we don't already have, then they will send it back. But if it's a species that we have already, they'll let it go. So if they can identify it as this is Duroceras reticulatum, this is the white milky slug, they'll let that shipment in despite having that snail, because we already have it. Which means that when I go out and sample, I might get lineages of this particular species of slug that are very different from one another, because they're coming from different parts of the world, and they're arriving and establishing themselves as different genetic lineages. It's like a fascinating, I don't want to, I'm hesitant to say natural experiment, because it's all human mediated. So I'm not sure that qualifies as truly natural, but this experiment that we have, this urbanization experiment, and all of the biodiversity that we have in that urban environment makes for a really interesting place to do research.
Yeah.
So anyway, sorry, I got off topic. What do I do? I would contact the agencies that are tasked with removal of invasive species, and California is very serious about that, and I'm sure Florida is as well, because of our crops. And there are agencies that are tasked with removing species and monitoring the places where they were to make sure that they are truly gone, or if they can't be eradicated, that they are controlled, which just means that there's few enough species, or excuse me, not so many individuals that it becomes a pest problem.
I had a question. It's about kind of the maybe it's not a methodology question, but so you have how many observations now? A lot, right? Oh, 30,
yeah, like 35,000 or so.
35,000 observations, and the number of species was somewhere in the 135 to 145 range. Do you feel like with that many observations that like you're pretty confident that's basically how many sort of species of land snails and slugs that there are in
like
LA?
Have we got them all?
Yeah, or do you feel like that's enough observations to be like pretty close?
Yeah, we probably plateaued to what we've got, but then there's the opportunity. We have two of the biggest ports in Southern California, two of the biggest ports in the world, or at least in the US, maybe not the world. And there are things coming in all the time, and it's and then there are likely many species that are not that just don't have observations, because not everybody's using iNaturalist. So like, for example, we found just this last week, there was an observation of a species that should not be in California. And I was talking to a colleague about it, and they said, well, that must be the first one in the county. And I said, it's the first that will be documented, but it's likely not the first. It's just, we don't know about those other populations because how would I know about them? So anyway, so that's the more people that use iNaturalist, I think it's really great for people if they're interested in knowing what the biodiversity is around them. But then it's also really helpful, exceptionally helpful for scientists, because we now have a greater ability to see and analyze all of these observations in this huge, expansive area. Yeah.
Give another question from Kathy. Do you have an idea of how many species made it there accidentally via their eggs by clinging to mud on a shoe or attached to an item transported overseas? How long are their eggs viable?
Yeah, that's a great question. Eggs I think in horticulture plants, I think that's a really common way for snails and slugs to come in. So when California gets plants from Florida or Hawaii or anywhere else, it's very possible that there are snail eggs in it, and it could be weeks that you could have viable eggs, and depending on the species, but easily 3 weeks. So that's a long time, and that's even enough time for someone to plant those plants, in their landscaping and not have any idea. Oh gosh, yep. The giant African land snail. So the question is, Lisa Acatina Felica. The giant African land snail, has it made an appearance in California? It has made an appearance at the airport in people's luggage. And I'm not sure how it was detected. It's possible it was a dog that smelled it, like a pest sniffing dog, or I don't know if it was considered suspicious, but anyway, there were several times that live giant African land snails, or gals, were brought into the Los Angeles LAX airport and confiscated. So far, I am not aware of any giant African land snails established in Southern California. Though sometimes on iNaturalist, there will be an observation. It's usually of a student who was given an assignment, like go out and make 10 observations for a school class, and they'll look up like snail, bird, and they'll look up a picture, and they'll just post the picture that they found on the internet as their observation. And then like on iNaturalist, there are ways of flagging what you consider is not a likely observation, or finding that observation is a photo from the internet, and you can flag it and remove it. So sometimes that happens, but the short answer is no. Thankfully, we do not have any populations of giant African land snail as have occurred in Florida, and we're trying to keep it that way, because they can be real big, real terrible pests.
One of the more compelling ideas to me in your talk was how you're describing how urban environments can have a species mix that is distinct to itself, or more comparable to somewhere far away than the surrounding areas, just because of how the species are introduced. Has there been enough data collection in the other places where people are doing these projects, in Seattle or Atlanta, to know whether, like, the kind of mix that you have in LA is actually analogous to sort of other cities, for the same reason, just because they're all I think you could do, we haven't done that, but you could pretty easily do that for any, just using iNaturalist data. Like, for example, I gave a talk to the Boston Shell Club, and I highlighted what species were most common around the city of Boston, and compared it to the species of snails and slugs that were most common throughout the city of Los Angeles, and they were like 90% the same. So it's like this, the characteristics of a city, and make it amenable for certain species to do well there. And because the bigger the city, and the more horticulture moves in, the more potted plants, the more trade, the more likely you're going to have these species from all over the world in these different places. Yeah. Well, we're certainly interested to see what we find once we start here. I mean, the Fort Myers area is of course urban. It's not LA. It's not Seattle. It's not Atlanta, but it's an interesting mix. And we're excited to get started with it. So
that sounds really fun. Really exciting.
Yeah, thank you for the model, and thank you for tonight's program, and thank you everyone for joining us. If you want to share this program with others, the recording of it will be up on our website within a week, and you can just share it from there. And thank you very much. Thank you, Jann. Have a good rest of your day and your evening.
Thanks, everyone.
And all right. Good night. Goodbye.